Today we're gonna have a delight to listen to Sabrina Dodge from the University of Córdoba in Argentina, who's gonna talk about peripheral and tumor immune signatures in advanced HR2 negative breast cancers. The floor is your Sabrina.
Thank you. Good morning, everyone. My name is Sabrina. I am PhD candidate from the National University of Córdoba in Argentina, and today I will present our work on peripheral and tumor immune signatures that predict response to immunotherapy in advanced HER2 negative breast cancer. HER2 negative breast cancer is an aggressive subtype with limited treatment option and poor prognosis. And immune checkpoint blockade, particularly anti-PD-L1 therapy, has improved outcome in some patients, but only a fraction truly benefits. This variability creates a need for reliable biomarkers to identify likely response before therapy begins. Our goal was therefore to improve treatment selection through immune-based predictive biomarkers. We analyzed 26 patients with advanced HER2 negative breast cancer treated with durvalumab and anti-PD-L1 antibody, and peripheral blood was samples collect longitudinally starting at baseline throughout treatment until week 28. And tumor biopsies were available at baseline for a subset of patients. In peripheral blood, we perform high-dimensional flow cytometry and CyTOF, characterizing major leukocytes population and functional markers to capture immune composition and activation states. In tumor tissue, we perform bulk RNA sequencing to analyze global transcriptional programs associated with response. We integrate immune cells frequency with clinical data and hematological parameters, including survival outcome and routine blood measurements, in order to build a predictive framework. This integrative strategy allows us to move beyond descriptive findings and toward clinically relevant prediction. We first perform a computational clustering and annotation of CD45 immune cells in peripheral blood. When we compare responders and no-responders at baseline, we found that no-responders show significantly higher frequency of Ki-67 Treg regulatory T cells, and in the same line, the monocytes PD-L1 positive and EGD positive naive B cells. Importantly, when we perform survival analysis, high frequency of Ki-67 negative Treg and PD-L1 monocytes were associated with worse overall survival. And this suggests that more immunosuppressive systemic environments at baseline might limit the efficacy of immunotherapy. We then integrate immune cells population data with biochemical parameters to evaluate their predictive capacity using the area under the curve. From this analysis, Ki-67 Treg, eosinophils, and monocytes emerged as the strongest predictors. And for each population, we determine the optimal cutoff, and we calculate false positive in dark blue and false negative in dark red. And individually, this marker had a low ratio of false result, but when we combine the three together, the predictive performance improved even further. As you can see here, Treg Ki-67 negative stand out as the strongest blood basis candidate for patient stratification in this treatment setting. Given the relevance of Treg in blood, we asked whether they were also important in tumor, and for that we analyzed publicly available single-cell RNA sequencing from a cohort of triple-negative breast cancer patients treated with the same therapy as ours, and focusing on baseline intratumoral Treg. We clustered the intratumoral Tregs in different clusters, and cluster 0 and 4 showed significant difference between responder and no responders. We then run a pseudo-time analyze, which is a computational method that orders single-cell alone trajectory based on their gene expression profile. And in these scales, values closer to 0 represent less differentiated cells, while higher values indicate more differentiated states. Using this approach, we found that cluster 0 was predominantly composed of less differentiated cells. When we looked at the gene expression profile of all cluster, we saw the cluster 0 expressed genes typically associated with naive-like phenotypes. So Treg in the tumor have different phenotype depending on the patient's response, and naive-like Treg are more abundant in responders. Here in the bulk sequencing, after observing phenotypic difference in tumor infiltrating Treg at the single cell level, we asked whether broader transcriptional program in the tumor microenvironment also differ between responder and non-responders. We first interrogated the immunological gene sets using C7 collection, and interestingly, no responders show enrichment of signature associated with regulatory T cell program. And this further support the central role of Treg associated immunosuppression in resistant tumor. And expanding our analysis to global gene ontology pathway, bulk RNA sequencing revealed clear transcriptional difference in between the two groups. In non-responder, we observe enrichment of genes signature related to extracellular matrix organization, tissue modeling, and muscle development programs. These pathways are often associated with stromal activation and matrix deposition, and suggesting the presence of denser and potentially more immunosuppressive microenvironments. Such remodeling may create both physical and functional barriers that limit immune cell infiltration and activity. In contrast, responders show enrichment of pathway related to mitochondrial ATP synthesis and electron transport change and oxidative phosphorylation. And this metabolic signature suggests that a more active mitochondrial program, which may support sustained immune activation and anti-tumor response. And emerging evidence indicates that mitochondrial fitness is critical for effective T cell function, particularly in the context of immunotherapy. Altogether, these findings suggest that response to immunotherapy is not determined solely by immune cells abundance, but also by the structural and metabolic state of the tumor microenvironment. In summary, in peripheral blood, at baseline, Ki-67 Treg and monocytes, PD-L1 positive and EGD-B cells are increased in no responders, and Treg Ki-67 negative and monocytes PD-L1 positive are linked to worse survival, and Treg Ki-67 eosinophils and monocytes are strong predictor, and their combination improve predictive accuracy. In tumor, in Treg phenotype differ between responders and non-responders. Responders have more naive-like Treg. And in bulk, RNA sequencing reveals distinct tumor microenvironment program in between responder and non-responders. For the next step, we want to expand our work by adding proteomic profiling serum and spatial tissue mapping in biopsies. And by integrating this data with the immune cells information we already have, our goal is to build an integrative biomarker model. And then we will validate our results in an independent HER2-negative breast cancer cohort, and hopefully move toward a biomarker panel that can be used in the clinical to give patient stratification for immunotherapy. And I will thank the organizers for giving the opportunity to present this work. I would like to thank our institute also in Argentina, Córdoba, and to our collaborator in Madrid, Spain, and Zurich, Switzerland, for making this project possible. A special thanks to the patient and their family who generously participate in this study, as well as to my lab group for their support, and thank you all for your attention.
Beautiful work, Sabrina. It's really nice to see you again. We have questions in the chat regarding how we use the Treg Chi67 info in like targeting or more like diagnostic, like targeting this population or just by diagnostic.
Yeah, we'll focus on Treg because our results in blood samples in peripheral blood is very interesting because in this moment we is a few biomarker from blood and is very important the blood marker because it's very easy the collection in comparison to the tissue or biopsies. We focus on the biomarker predictive for blood samples because it's permission that predictive and then also monitoring in a long time. So we focus on the different immune population and we found the Treg. So we study a little bit how if is it possible use for for predictive, but also study this role of Treg on the tumor. So we will focus now on the blood sample and connection with tissue and search if it's possible integrate the both parts.
Thank you, Sabrina. We have a couple of questions regarding the phenotype of the Tregs that are infiltrated in the tumor. Particularly, Luis asked, what do you think that restrained the naive phenotype Treg population that you found in the infiltrating the tumors? Why they are not activating?
Yeah, maybe here we can found for us also was very interesting the Treg light naive, and the more interesting was the in the tumor have different phenotype of Treg, and why what is the different? Our goal was what is the different in between the Treg that have the responder patient and the no responder patient, and about the phenotype we can found some difference in the phenotype and maybe could be less activation Treg. So if they don't activate in the tumor, this relation in between CD8 cells, who the CD8 T cells is kills to the cancer. So if have less activation, we suppose that have the good relation to the 8 CD8 can kill to the cancer cells, you know. And this is our thinking, but we need to have some studies about the activation to this Treg for that. But we think that it's about the activation. Maybe it's less activation.
Yeah, just I guess one more question from Eleanor as well. Do you think these phenotypes of the Tregs could be shared between all the tumors, at least for the mammalian tumors, or it's just a particular of this the tumor? Have you looked at other type of tumors different from HER2?
Yes, we couldn't study in this time other tumor or the Treg how is in other tumor different, but it's our goal because we will integrative this algorithm integrative for first for breast cancer, but then we will focus on the repeat these results in other tumor, but now is not the script the Treg naive like in other tumor is not so much. But yes, we could found some studies in other like melanoma, other cancer, but we will focus after that about the study in other tumor and if these biomarkers would be extrapolated in other tumor. Yes, this is our next step.
Thank you so much, Sabrina. I think there are some more questions in the chat if you could help us with that. In the interest of time, we will continue with the next talk now from
Perfect. Thank you.
Mohammed. Yeah, thank you.